I'll be doing a presentation on meiosis. It's going to be a video format, which will be my new edition. On this presentation is actually meant to be our revision material as we prepare for the upcoming examination. So here is a topic overview on what to expect. This topic involves a lot of labeling of diagrams, functions to state, drawing of specific structures, identification of processes, and then we have description of processes. And in exams, if you are required to describe, it means to write the steps involved. No explanation is needed. So I'll begin by focusing on the revision of the structure of a cell with emphasis on the following parts. We have the nucleus, the centrosome, and the cytoplasm. So those are the main parts that we need to know as we move forward. The functions of these parts and other parts are required for examination purposes. So here are the following parts that are required for exams. We have the nuclear membrane. It encloses and protects the DNA, and that is a function. And then we have centromere. It joins the two sister chromatids together. We have the cell membrane. It splits the cytoplasm into two during cytokinesis. And then it also encloses and regulates the movement of substances. What then is cytokinesis? It is the splitting of the cytoplasm, and it occurs during telophase I and II. We have centrosome. It forms the spindle fibers. It is actually an organelle that forms the spindle fibers. What are the functions of the spindle fibers? They contract and pull chromosomes or chromatids to the opposite poles of the cell during anaphase 1 or 2. You are hereby advised to memorize these functions very well since they forms the bedrock of the topic meiosis. The entire concept of meiosis rests on these special component of a cell, and they are required for examination purposes. The next question we're going to try and find an answer to is, what is a chromosome? So that is a diagram. You have it on your screen. A chromosome is a thread-like structure made up of DNA and is found in the nucleus of each cell. They are passed from parents to offspring. In other words, you only get it through inheritance. They are packaged together with the special proteins called histones. So that is a diagram of a chromosome. It consists of two chromatids joined together by a structure called the centromere. The sister chromatids are identical to each other. One thing we need to know also is that chromosomes are made of DNA, and DNA contains the specific instructions that make each type of living creature unique. Well, we have on our screen an interesting illustration. Over here, we have two chromatids, and they already have their centromere. But then you realize that under the process of replication, they changed to now appear as the letter X, and the two look very much alike. Hence, we call them homologous chromosomes. If you examine further, if you look previously, we had only one threaded chromatid, but then Here we are, we have two threaded chromatids for each chromosome. So the question is, how did this change to become like this? And the answer is simple. Chromosomes, which are single threads, become double because of DNA replication. In other words, DNA replication converted the single threaded chromatids into double threaded structures, what we call chromosomes. And we need to also know that the two chromatids, after DNA replication, are then joined together by a special structure called centromere. We should also bear in mind that chromosomes are made up of DNA, and from time to time, during cell division, DNA undergoes the process of replication to make identical copies of itself so that they can be shared by the new delta cells that are formed as a result of cell division. Now we are going to do something special. We are going to look at this process in reverse. So we'll be dealing with replicated and unreplicated chromosome. So on your screen, we have our homologous chromosomes. Those are two sets of chromosomes. They look very similar if you check the length, the size, and appearance, except the color. They have almost everything in common, not the same. But then, if you check carefully, this is what we call a replicated chromosome, as we saw earlier on. On our right side of the screen, we have two sets of homologous chromosomes again, but however, they appear as single threads. We call them unreplicated chromosomes. Now, you want to find out, what do you think could have caused the replicated chromosome to change into an unreplicated chromosome? Is there a process or a mechanism that can cause replicated chromosome to change to become unreplicated chromosomes? Of course. And it is actually anaphase 2. What goes on during anaphase 2? During anaphase 2, spindle fibers contracts and pulls chromatids, causing the centromere to split or to break. This causes sister chromatids to move to the opposite poles. So the reason why a replicated chromosome will change and become an unreplicated chromosome is due to the event that occurs during anaphase 2. So the reverse of the previous process is actually when replicated chromosome through anaphase 2 changes and then becomes unreplicated chromosomes. Same can be said of this. Here we have an unreplicated chromosome changing to become a replicated chromosome changing to become an unreplicated chromosome. And the only mechanism that can make that possible occurs during anaphase 2. And one more time, spindle fibers contract and pulls chromatids, causing the centromere to split, that is, to break, and this causes sister chromatids to move to the opposite poles. Now let us study the structures of chromosomes. And this is something we already know. And as a revision tool, we need to know something about the structure of chromosomes. Chromosomes are made up of DNA and protein, a special kind of protein called histones. The number of chromosomes in a cell is characteristic and unique of an organism. That is, in humans, there are 46 chromosomes. You can't find the same in housefly. In housefly, you will find only 4 chromosomes. In the case of other primates like chimpanzee, they have 48 chromosomes. The number of chromosomes differ from one organism to another. Well, on your screen, we have two sets of chromosomes, like we saw earlier on. If you examine them carefully, they look very similar. If you just look at the length, their size and appearance, they are almost identical except their coloration. They have different colors. Now we need to provide an explanation for what you are seeing on the screen. So the question we're going to ask is, what are homologous chromosomes? Because you can see they are already labeled homologous chromosomes. So what are they? Why are we saying these two chromosomes are homologous? So the reason why we are saying they are homologous is because they are similar in length. As you can see on the screen, the two chromosomes have similar length, not the same. They are also similar in size. As it appears there, their thickness are very similar. And they also have the same gene locus. That's the position of their alleles are almost the same. Okay. But then if you look carefully, you may wonder why they're not appearing in the same color, even though they are similar in length and size, and they are always in pair. Why they're not having the same color? And the reason is simple. It's because normally one is inherited from the mother and one is inherited from their father. So anytime you find or you come across homologous chromosomes, you have to know they won't appear the same way. In most exam question papers, one is represented as white, the other one is represented as black. In other textbooks, you can use different colors to represent them. The two colors must never be the same. And the reason is because one is actually a paternal chromosome belonging to the father, and the other one is a maternal chromosome. So it's the reason why they appear the way they do, and they are always in pair. Well, let us explore the terminology karyotype. What is a karyotype? A karyotype is a representation or a display of the number, shape, and arrangement of a full set of chromosomes in the nucleus of a somatic cell. A karyotype is a representation or display of the number, shape, and arrangement of a full set of chromosomes in the nucleus of a somatic cell. Well, so on your screen, we have here a human karyotype, and you can see they are all given numbers. The numbers represent the position occupied by the pair of chromosomes or the two sets of chromosomes. At position 1, we have two chromosomes. If you go to position 5, we also have two chromosomes. Now, the chromosomes at position 1 and position 5 and position 13, they don't look alike. But then, in each position, if you look at position 1, the chromosomes there are identical. If you go to position 10, the chromosomes there, they look similar. Same applies to position 19. The chromosomes are all similar. Now, what we need to know is that homologous pairs of chromosomes in a human being forms what is called the karyotypes, and they are displayed in a diploid cell. Diploid means the cell consists of the full sets of chromosome for that particular organism. And since we are dealing with human karyotype, the full sets of chromosome that will be displayed will be 46. Now, like we saw earlier on, homologous chromosomes are similar in many ways. Among the many ways, these are the major ways in which they are similar, as they are arranged in a karyotype. They are all similar in size. That is, if we look at chromosome position number 8, the chromosomes there are similar, but then the chromosomes at position 8 are different from those at position 9. So the chromosomes have arranged themselves in pair according to their similarity in size and also their similarity in length. We should note that These chromosomes are not the same. They are not 100% identical. They can only be at most similar. Now, if you look at position 23, which is not labeled, you realize that there is something special about them. If you look at chromosome 1 to 22, mostly they are very similar. You have two similar chromosomes occupying the same position, except position 23. In females, it is discovered that at position 23, the chromosomes there appear large and identical. In human males, it is also observed that at position 23, the chromosomes there they are not identical. One is large and one is small, and that is how they are in human males. These chromosomes, due to their uniqueness, they are called gonosome. We're going to explore what gonosomes are as we move forward. Well, we are still on karyotype. It is a display of the number, shape, and arrangement of the chromosomes in the nucleus of a somatic cell. These karyotypes that you are seeing here are usually displayed during prophase I, when homologous chromosomes pair up. So if you look carefully, from chromosome 1 to 22, they are called by different names, except chromosome 23, where in human females, they appear identical, but then in human males, they appear strikingly unidentical. These chromosomes are involved in the determination of sex. Sex here means gender. And because these chromosomes are only involved in sex determination, they are therefore called sex chromosomes. The word sex here means gender. The rest of the chromosomes from 1 to 22, they are not involved in sex determination. They are involved in any other phenotype of an organism, any other physical appearance of an organism except the gender. They can contribute to the formation of the gender, but they do not determine whether an organism will be a male or a female. Hence, they are called autosomes. Sex chromosomes, on the other hand, are sometimes called gonosomes. So that is the term that applies to chromosome number 23. So we'll be looking at sex determination and inheritance of sex. The word sex here represents gender, and there are two kinds in the sciences, in the genetics. So in humans, if you take the human karyotype like we saw earlier on, chromosome 23 is involved in sex determination. Hence, we call them sex chromosomes or gonosomes. Like I said earlier on, it is observed that in females at position 23, there are two chromosomes and they look very similar. They are homologous. In human males, there are two chromosomes, but they are not homologous, meaning they are not similar. Mostly one is large, the other one is small. And if the chromosome at position 23 are both large and similar, that person becomes a female. If the chromosomes at position 23 are not identical and are not homologous, that is, one is large and one is small, then that person becomes a male. And it's the reason why we classify the chromosomes at position 23 as sex chromosomes, or we also call them gonosomes. Well, so how do you describe in exams if somebody is a male or female by observing or by looking at chromosomes at position 23? So here is how you approach it. That is inheritance of sex. XX is for female, XY is for male. But for examination purposes, this is how you approach it. After meiosis, an ovum will have 22 autosomes and an X gonosome. A female will therefore have two large identical or X gonosomes, like you can see on your screen. After position 21, we go to position 23. Here, the person can either have two identical gonosomes or two X chromosomes if it is given to you in exams. If the letter X is not given, you then say the person is a female because she has two X chromosomes. No, you don't say that. You will say there are two large identical gonosomes. Secondly, a male will have two unidentical gonosomes, like it is displayed there, or an X gonosome and a Y gonosome at pair 23. And that is how you explain it in exams. Be careful with the use of XX and XY to explain a gender. If it is not given to you in exams, you don't use those letters. You simply say, if the person is a female, you explain by saying, give the reasons by saying, this person is a female due to the presence of two large identical gonosomes. You don't say XX chromosomes are present at position 23, whereas it has not been given to you. But if it is given to you, you can simply say there are two X gonosomes there. So you have to be very careful when you're approaching some of these things. And I'll be laying much emphasis in class. Now, there is a simple analogy in the form of an illustration here. We have, let's start with the first one. So we have two different people here. We assume they are twins. We have A and B. We are supposed to prove if they are identical twins. Identical twins have the same genders. Okay, they have the same sex. Unidentical twins do not have the same sex. So let us find out if A and B are identical. If A and B are identical, they will have the same sex. So let's start with A. So to identify the gender or the sex of a person, you have to go to uh, position number 23 by observing or studying the karyotype of the person. So at pair 23, you would observe that it has been given to you already in the question. There are two unidentical gonosomes there. One is large, one is small. And it has been labeled for you. There is one X gonosome and one Y gonosome. If you compare it with individual B at pair 23, at position 23, there are two X gonosomes present there. Okay. And by interpretation, if you look at, if we observe it further, you realize that their genders or their sex are not the same because they don't have the same gender. A is a male, B is a female. We say they are unidentical twins. Whereas if you check the other situation here, we have two twins, twin A and twin B. We are supposed to prove if they are identical twins or not. And if they are identical twins, they will have the same gender. So if you check carefully, if you go to position 23, which is labeled X for you, you realize that the chromosomes there, they are very identical. There are two identical gonosomes there labeled X gonosome. If you check twin B, same thing applies there. And that means both twins have the same gender. Because their sex are the same, we say twin A and twin B are identical. Whereas on the left side, A is a male, B is a female, hence they are not identical. They don't have anything in common. They are very unique. So if you go back to it again, let us identify the sexes. Let's label them. A is a male and B is a female. Why is A a male? Because at position 23, uh, there is one X gonosome and then there is one Y gonosome. Or you can say the gonosomes are not identical. One is large, and one is small. If the letter X and Y is not given to you, don't predetermine that it's going to be X and Y. You only state that it's X and Y if it is given to you in the question. If you check B, B is a female because there are two chromosomes there and they are both identical. They are both large. Now you don't have to say there are two X chromosomes there. But then you can say so in this particular situation because it has been given to you in the question. So here you can say at position 23, there are two X chromosomes. But be careful in its usage. If it is not given to you, do not decide. So if you check on the other side, okay, on the right side, twin A is a female because of the same reason. There are two large X chromosomes there, or two large chromosomes there. And then if you check the second one too, it's a female because there are two large chromosomes. And because of that, on our left side, these twins, okay, we need to know what they are. Remember, I said identical twins, they have the same rod, they have the same sex and gender. But then if you look, if two twins are identical, we can say they have the same gender. But as you can see on the screen, these twins are not identical. So we can say they don't have the same gender, hence we don't say they're identical. But then if you check those here, they are identical twins, and the reason is because they have the same gender. All right, so um that is that. Uh All right, so let's study the situation here. Let's start with the one on the left. We have two individuals, and we are supposed to prove if they are twins or not. But here is the clue. If two twins are identical, they will have the same gender. If they do not have the same gender, we say they are unidentical twins. So, from our previous study, we can actually use the karyotype to determine only two things. One is to determine any genetic disorders, more specifically Down syndrome, or to determine the sex or the gender of the individual. So let's give it a try. Let's identify the gender for individual A. Try and identify the gender for individual A. And to do so, you have to go to pair 23, which is not labeled. But then if you count it well, that is position 23 on the karyotype. And over there, you will discover that there are two chromosomes there. One is large, one is small. The large one is labeled as X, the smaller one is labeled as Y. Let's check with individual B. At position 23 also, you will discover that there are two gonosomes there. Both of them are large and both are labeled as X, or they are both labeled with the letter X. Now, let's go to the other side of the video here, or on your screen, sorry. So over here they are labeled as twin. We have twin A and twin B. That means we are dealing with twins here. So, like I said earlier on, if the twins are identical, they'll have the same gender. If they're not identical, they'll have different sex. So let's determine if twin A and twin B are identical or not by checking their sex. So we go to position again one more time. We have to go to position 23 on the of the karyotype of twin A, and over there you realize that the gonosomes there are both large. If we go to twin B also, we have the same situation. At position 23, the gonosomes are also large. So, what can be the conclusion here? To draw the conclusion, we have to interpret and then depict the phenotype for this individual. So, this individual is a male, and it's due to the simple reason that at position 23, there are two gonosomes there. One is large and one is small. Here it is given to us in the question, whereby the large one is labeled as X, the smaller one is labeled as Y. Hence, we can say at position 23, there are two chromosomes, one is X and one is Y. And the reason I'm saying X and Y is because it's given to you. Because If you check under a microscope, you don't see X and Y there. You see one large chromosome and one small chromosome. The large one is assumed to resemble the letter X. So be careful when you are answering question on sex determination. Don't assume it is always X and Y unless it is given to you in in the exam question paper. Like it is on your screen, it has been given to you. So you are free to say at position 23, there are two chromosomes, one is X or one is Y. But the most accurate way of answering it is to say at position 23, there are two chromosomes, one is large and one is small. If you go to twin B is a female. What are the reasons? Because again, at position 23, there are two chromosomes and both of them are large. And that is it. It ends there. So, by interpretation, we can say here also for twin A that twin A is a female. Why? This for the same reason, being that at position 23, there are two large gonosomes. And then the same thing applies to twin B, the same reason at position 23, there are also two large gonosomes. In this particular situation, these gonosomes are labeled as X. So you are free to say at position 23, there are two X chromosomes because it is given to you. But if it's not given to you, you don't give such an answer. You simply say at position 23, there are two large gonosomes and it ends there. So let us complete our interpretation and draw conclusion. So here we can say individual A and B are actually what? Unidentical twins. Why are they unidentical twins? The simple reason is because they do not have the same sex or gender. One is a male, one is a female. Hence, they are not identical. On the other side of the coin, twin A and twin B are identical twins, and the simple reason is because they have the same sex or gender. All right, now let us explore some important terminologies which are required in the study of meiosis. So we start by looking at the word haploid. What is the meaning of the word haploid? Usually represented with the letter N. It refers to cells that contain only half the total number of chromosomes. Example, the total sex or number of chromosomes in a human cell is 46. Of all cells in a human body, only sperm cells and the ovum of humans contains 23 chromosomes, which is half the total number 46. So that is the reason why. Hence, we can say the condition or the state of the human sperm cell or ovum with 23 chromosome is called haploid. So the word haploid is always stated in terms of number of chromosomes. And the number of chromosomes differ from organism to organism. The other word connected to haploid is the word diploid, represented numerically as 2n. It represents cells that contain two sets or complete number of chromosomes in terms of chromosome number. Example, every cell in the human body except sperm cells and ovum contains 46 chromosomes. So every cell means all the cells in the human body except our sperm cells and ovum in females. So in other words, the condition or state in which a muscle cell has 46 chromosomes is called diploid. E.g., example, the human skin, hair or muscle cells all have 46 chromosomes in their nucleus. And this is something we should know. So the keyword, the two keywords here are haploid and diploid. And these are words that are used in terms of chromosome number. Now let us try and play along with it because we have to know diploid and haploid represent the condition or state in which a cell is. Let's do a simple analogy here to test our understanding. Now we have here sex cell, that is gametes, and different kinds of it. We have different types of it. And then we have haploid, And we have somatic cells, and then we have diploid. We are supposed to know clearly that all sex cells are also called gametes, and all gametes are automatically haploid. On the other side, all somatic cells, all means all somatic cells are also called diploid. So we have examples of sex cells, and then we have examples of somatic cells in different organisms. But then there are two types of sex cells, sperm and an ovum. So let's start by looking at this. Let's say in the human sperm cell, there are 23 chromosomes, and that is actually the case. How many chromosomes will be found or will be there in the human muscle cells? In the diploid state, there will be 46. So give it a try here. In a chimpanzee hair cells, there are 46 chromosomes there, making it diploid. How many chromosomes will you find in the nucleus of a chimpanzee sperm cell? It's going to be half of it because haploid sounds like the word half. So half of 48 will be 48 will be 24. Now let's also look at plant ovules and plant cell on the leaves and plant leaf cells. If there are 18 chromosomes in plant ovules, ovules represent the gamete, and if it is haploid, then the leaf cells, which are somatic and automatically diploid, will have how many? The word di means two. In this particular case, we're looking at times two. So if there are 18 chromosomes in the plant ovules, then it means there will be 36. So we can continue by trying the others. There are only 11 haploid chromosomes. We have 11 chromosomes in the haploid cell, that is the pollen grain. How many chromosomes will be there in the root hair cells? That's going to be times 2, 22. What about if we have 4 chromosomes in the body cell of a housefly? How many chromosomes will you find in the sperm cell of a housefly? It's going to be 2. 2 means haploid, that is half of 4. What about dogs? In the case of their bones, you'll find 78 chromosomes. What about their sperms? How many chromosomes will you find there? 39. That is half the number of the diploid. Half of 78 is 39. What about the ovum of leopard frog? If there are 13 chromosomes in the ovum, how many will you find in the skin cell? Remember, the skin cells are also somatic cells and they are automatically diploid. Di means times 2. So 13 times 2 give us 26. So that is the analogy, and that is how we approach haploid and diploid. All right, so we are still on the basic terminology. What are sex cells? Sex cells are also called gametes. They are the reproductive cells of an organism. Example, we have sperm cells and ovum, also sometimes called egg cells, but then the correct word here is the word ovum. What about somatic cells? Somatic cells too are called body cells. They are the cells that forms the body of an organism. With regards to multicellular organisms like humans and animals and plants, okay, the cells that forms their body or our body is called somatic cell. The word soma means body. Hence, somatic cells are also called body cells. Now, let's look at the word gonosomes. So, what are gonosomes? Gonosomes are also called sex chromosomes. They are a type of chromosomes involved in sex determination. The word sex here means gender. So females have two large gonosomes represented with the letter X. Hence, we can say they have two X chromosomes. Males have two gonosomes. One is large, one is small. The smaller one is represented with the letter Y, and the large one is represented with the letter X. So males have one X and one Y chromosomes, whereas females have two X chromosomes. Right. What are autosomes? Autosomes are any chromosomes that is not involved in determination of sex in an organism. So on your screen, we have chromosome 1 to chromosome 22. These chromosomes are not involved in sex determination. Hence, they are called autosomal chromosomes, or simply put, autosomes. But then, position 23, there are also two sets of chromosomes there, and they are involved in sex determination. Hence, we call them sex chromosomes. Sometimes they're identical, sometimes they are not identical. If they're identical, the person will become a female. If they're not identical, the person will be a male. Well, we are back to the terminology again, karyotype, and I hope you haven't forgotten, because it means, or it represents the display of the number, shape, and arrangement of a full set of chromosomes in the nucleus of a somatic cell. All somatic cells are automatically diploid cells. So that is a karyotype of humans, and they are actually labeled or given positions, starting from 1 to position 23. Position 1 and 22 are involved in the determination of the body, or the body of the organism. That is mainly the structural appearance of the organism. Whereas position 23 deals specifically with the gender, whether the organism will be a male or a female. Take note of it. So if you look carefully, all the chromosomes in this bracket, that is chromosome 1 to chromosome 22, because they are not involved in sex determination, they are called autosomes. But then after position 22, we go to position 23. Remember, there are 46 chromosomes there. And at each position, there are two chromosomes. So chromosome number 1 to 22, which represent 44 chromosomes, are what we call the autosomes. Now, at position 23, the chromosomes can sometimes be identical, or they may not be identical. If they're identical, we represent them with two letter X. If they're not identical, we represented them using one letter X and one letter Y. And these chromosomes are involved in gender determination or sex determination. Hence, we call them sex chromosomes or gonosomes. And there are only two chromosomes that are involved in sex determination. Hence, there are two gonosomes here, but then there are 44 autosomes. So altogether, there are 46 chromosomes. Now, you may have to try the following to test your understanding and your memory strength by drawing the structure of a chromosome and label the centromere and the chromatid. Then differentiate between haploid and diploid cells in terms of chromosome number. And then differentiate between sex cells, also called gametes, and somatic cells, also called body cells. And then also differentiate between sex chromosomes, that is gonosomes, and autosomes. You can just give it a try, and later on, go and check through your notes and find out how best you're able to remember and how strong your memory power is. Lastly, define the word homologous chromosomes. So, what are homologous chromosomes? Try and also brainstorm on it, crack your head on these particular terms. These are what we have covered so far. All right, so we'll be moving on to the other aspect of meiosis, where we need to know what meiosis is properly. So, what is meiosis? It is a type of cell division whereby a diploid cell, into brackets somatic cell, undergoes two cell divisions and divides to form four dissimilar. Another word for dissimilar is the word unidentical haploid cells. And all haploid cells are automatically called sex cells. In other words, meiosis leads to the formation of sex cells or gametes. And know there are two types of gametes, sperm cells and ovum. Now let us look at the site or the location of meiosis. So, since meiosis is a cell division that usually takes place in sex cells, uh, these sex cells are also called gametes or They are also sometimes called reproductive cells. In plants, meiosis occurs in the anther and it occurs there to produce pollen grains. In the ovary, it occurs there to produce ovules. In other words, in the male part of the plant called anther, meiosis occurs to produce pollen grains, whereas in the female part called the ovary, it occurs there to produce ovules. What about humans? In humans, meiosis occurs in the testes of males to produce sperms. And then it also occurs in the ovary of females to produce ovum. Now let's move on to the process of meiosis. So you are required to revise the process of mitosis from grade 10. You may therefore take a few minutes or you can freeze the screen and go through the process and remind yourself of of the phases of meiosis from grade 10. Remember, there are four phases of mitosis. We have prophase, metaphase, anaphase, and telophase. Take your time and read through the events under each phase, and then continue uh, watching the presentation. All right, so my focus will be on meiosis because that is uh, what that's the main area exam questions will be focused on, will be concentrated on. So we started by looking at the definition, and we discovered that once again, it's a type of cell division where a diploid cell, also called somatic cells, undergoes two cell divisions and divides to form four dissimilar or unidentical haploid cells, also called sex cells. And we also looked at the site of meiosis, and we said it occurs in sex cells. And in plants, meiosis occurs in the anther to produce pollen grains, and it also occurs in the ovary to produce ovule. In humans, meiosis occurs in the testes to produce sperm cells, and in the ovary to produce ovum. So here we are on the screen. We are looking at the phases or the process of meiosis in a nutshell. So with meiosis, you start with one cell, and then at the end of the process, you end up with four daughter cells that are not identical. These four daughter cells are haploid and they are automatically sex cells. They will develop into gametes. In males, they will develop into sperm cells, and in females, they will develop into an ovum. In meiosis, you start with more chromosomes and you end with half the number. So in your on your screen, we started with before meiosis one, we started with four chromosomes. At the end of meiosis, we only have two chromosomes. Yes, and one more time, the number of chromosomes you start and end with can vary from organism to organism. So over here, we started with two. In our previous video, we started with four. Here, since we started with two, at the end of meiosis, we'll end up with only one chromosome in each cell. But the following remains the same. The number of cells we started with will remain, the number of cells we started with won't remain the same. At the end of it, we're going to end with four. So here we started with only one cell. At the end of it, we have how many? Four cells. And these cells are not identical. But then we started with two chromosomes. You're going to end with only one chromosome. As compared to our previous video, we started with only four chromosomes. We ended up with two. But then in our previous video, as you saw earlier on, we started with only one cell. We ended up with four cells, which are not identical. Same applies to this particular process too. It doesn't matter the number of chromosomes you start with. You always start with one cell and you are going to end up with four cells. And those are the things you should pay attention to. All right, so we have meiosis and mitosis on our screen. We need to understand that our focus will be on meiosis. So here, instead of drawing chromosomes, we have represented the number of chromosomes using a numerical value. So 2n means diploid, 1n means haploid. That is to say, during meiosis, you start with a diploid cell, which is 2n, and you end with a haploid cell, which is 1n. 1n means it is haploid. It is half the number of chromosomes you started with. Hence, if you started with 10 chromosomes, you are going to end with 5. If you started with 4, you're going to end up with 2. If you started with 8, you're going to end up with 4. However, it doesn't matter how many number of chromosomes you started with. The number of cells in all the processes remain the same. That is, you always start with one cell. You're going to end with 4 cells. As compared to mitosis, you start with diploid cells and you end up with diploid cells. You start with 2n, you end up with 2n. You start with one cell, you end up with 2. And mitosis is only involved in growth, whereas meiosis is involved in gamete formation, also called gametogenesis. So let's look at the process of meiosis. Now, the process of meiosis consists of two cell division. The first one is called meiosis 1. It starts with interphase. During interphase, DNA replication takes place. And then the next phase is prophase 1. You must always give it a number. It is either Prophase I or 2. So since you are dealing with meiosis 1, this is going to be Prophase 1. Now, this phase includes the description of crossing over, which is very important. Metaphase 1 deals with the random arrangement of chromosomes. And then we have anaphase 1, which we are going to look into, and then we have for telophase 1. The most important detail have been listed on your screen for interphase, prophase, and metaphase 1. Then after telophase 1, we go to the second meiotic division, which is meiosis 2, and the process repeats itself with some few exceptions and differences. So we have prophase 2, this time no crossing over. Then we have metaphase 2, and this time there will still be random arrangement, but then there will be a slight difference in the number of chromosomes that will actually arrange themselves. And then we have anaphase 2, and then we have telophase 2. So that is the process. It is actually two meiotic division. And do not forget in exam, if you have to answer a question and you have to, if let's say you have to identify a particular phase, do not forget to specify. If the answer is anaphase, make sure you tell us the number, whether it's anaphase 1 or anaphase 2. Do not just write anaphase without giving us the number. If the answer is anaphase 1 and you wrote anaphase, you won't be marked correct. So make sure you specify. Under each phase, it is either 1 or 2. All right, so in looking at the process of meiosis, we are going to uh, start with the process of interphase. So what goes on during interphase? Events of interphase. So during interphase, DNA replication takes place. That is all you need to know. And then you need to be able to describe what goes on. During interphase, chromosomes which are single threads become double. That is, each chromosome will now consist of two chromatids joined by a centromere. And then we need to also know that DNA replication helps to double the genetic material. And why should that happen? Why should the genetic material double itself? It does so that it can be shared by the new cells arising from the cell division. So there's a reason why DNA replication uh, occurs during interphase. All right, so let us examine the following. Over here on our screen, you remember this diagram we met earlier on where an unreplicated chromosome, uh, through the process of gene replication, became replicated. So there is something you need to pay attention to, which is actually going to help you when you are approaching examination questions on meiosis. Anytime you see a diagram with this, with chromosomes appearing like this, okay, you have to know you are actually looking at telophase II, and it is actually the end of meiosis. And at the end of meiosis, chromosomes usually appear as it is on your screen. They appear as single threads. Okay, they will appear as chromatids. So whenever you see such, you come across a question with such a display, a diagrammatic display of chromosomes within a particular question, you have to know you are actually looking at telophase. There are some other vital techniques I'll be sharing with you directly in class. This is just for a revision purpose. All right, so let's just look at each phase, but I'm not gonna delve deep into it since it's just a revision exercise. Much of the work will have to be done by you. You have to memorize each phase of meiosis, all the 8 phases. You need to be able to memorize it. But I'm gonna actually touch on some key important points in each phase. Okay, but I'm not gonna go so much into it. So under Prophase I, we are supposed to be able to describe the process of crossing over because that's the most important event that occurs during Prophase I. So let's look at crossing over. Whenever you see a diagram like this, you see a display of chromosomes behaving this way. You have to know that you are actually looking at Prophase I, and what is happening is what we call crossing over of homologous chromosomes. And you must be able to describe it as it is stated in the examination guideline that you need to include the description of crossing over. Description means you have to write it as it is. So let us look at crossing over event. It occurs during Prophase I, and homologous chromosomes forms pairs or bivalent, that is, they stand side by side, and then adjacent chromatids of each bivalent touch each other at a point called a chiasma. So, like you see in the second display, the first one you see chromosomes are standing side by side. The second one you see the chromosomes have actually overlapped. The point of overlap or the point where they touch is what is called chiasma. And they do so that chromatic segments can break off and then they are actually swapped. So at the end of crossing over, what's going to happen is that this event is going to result in the exchange of genetic material. As you can see there, now on your screen, we started with an all red and an all blue chromosome. But then at the end of the process, there is a mixture of colors. Once you see a mixture of such a color, of such a display in any textbook, you have to know that crossing over has already occurred. Your exam papers are actually printed in black and white. Hence, the display is going to be a chromosome, which is part white and part black. And once you see such a display, you have to know that crossing over has already occurred. And you are not actually in Prophase I, you are in another phase. But then if you see a display as it is on your screen, if you see two chromosomes standing side by side and they are of uniform color, that is all white and all black, then you have to know that crossing over is about to occur. If you see two chromosomes, all black and all white, and they have touched each other or they have overlapped as it is in the middle, and as displayed in the middle diagram, then you have to know that crossing over is occurring. And these are some of the techniques you should know. So we need to know that the point on a chromosome at which chromosome occur is what is called chiasma. Chiasma is a terminology you need to pay attention to. So the main theme here is that crossing over occurs during prophase I, and it results in the exchange of genetic material. So remember what we studied earlier on. At the beginning, during interphase, we said chromosomes which are single threads become double, that is, two chromatids joined by a centromere, and as a result of DNA replication. So let us try and explore the following. Why Let us look at why homologous chromosomes are not identical. Uh, Anytime they are drawn, one is black and one is white, or one is given a different color and the other one is also given another color. What? Why is that the case? Why are they not identical? And the reason is because homologous chromosomes are different. And the simple reason is because one of the pair contains paternally derived genes and the other contains maternally derived genes. If the statement is a bit complicated for you, I've made it easier here. You can say, we can also say, put the answer in a different way. The reason is because homologous chromosomes are different simply because one of the pair contains genes from the father and the other one contains genes from the mother. I think the latter answer is more reasonably understood, and you can actually appreciate the way it is actually constructed. Then, why? Let us ask ourselves: why should chromosome actually undergo crossing over? Is it even necessary? Hence, we need to look at the significance of crossing over. So, crossing over is important because it introduces or it brings about variation within the population of an organism due to the exchange of genetic material. Mind you, meiosis leads to the formation of ovum and sperm cells. And what do you do with an ovum and a sperm cell? You fertilize it. And when you fertilize it, what do you form? You form a zygote. A new organism will emerge. Okay. And remember, during the process of meiosis and gametogenesis, crossing over does okay. It's the reason why in one single family you have siblings who do not look alike, even though they are all born from the same mother and the same father. They came from one mother and one father, yet they don't look alike. They all look different, and their differences can be traced to two major causes. There are actually other causes, but then for curriculum's sake, there are two major reasons why siblings don't look alike. And one reason is because of crossing over that occurred during gametogenesis, or crossing over that occurred during meiosis, most specifically Prophase I. I'm using gametogenesis here because meiosis causes gametogenesis, and meiosis leads to the formation of gametes. And the gametes here are sperms and ovum that will fertilize to produce another offspring, a zygote. I'll be saying it here. So the reason why siblings don't look alike is because of one reason, one of the many reasons being an influence from crossing over, because it introduces variation. What is variation? Variation here refers to the differences that exist among individuals of a population due to the differences in their DNA sequence. If you remember earlier on, also in my presentation, it was stated that chromosomes are made up of DNA. And if you remember from the concept of genetics, the DNA is made up of genes. And the total genetic makeup of an organism is what is called a genotype. And a genotype determines the phenotype. Hence, if there is crossing over, if there is a mixture or an exchange of genetic material between two chromosomes, then it means that the gamete that will be produced will actually consist of different combinations of genotype producing a different phenotype. And all these can be traced to crossing over. So as it is stated down there, variation is the differences that exist among individuals. And these variations are introduced or were introduced by crossing over. It was introduced through crossing over. So we need to explore deeper and find out which other meiotic phases, apart from Prophase I, which other meiotic phases introduces genetic variation, and that phase is going to be metaphase 1 and metaphase 2. What goes on between metaphase 1 and metaphase 2? There is a random arrangement of chromosomes. This random arrangement of chromosomes brings about further variation. Already Prophase has introduced variation. Metaphase will further bring about variation. That is, it will bring in more differences. And the reason why there is variation via metaphase 1 and 2 is because the chromosomes do not arrange themselves by predetermination. In other words, you don't determine, or nobody determines how the chromosomes should arrange themselves. They do so randomly. Randomly means they are not prearranged or they are not predetermined. Nobody tells which chromosome should arrange with what. They do so on their own accord. They do so randomly. Randomization brings about these differences. So, in a nutshell, we have to know that here are the mechanisms that introduce genetic variation within a population. Here we're looking at mechanism, not phase. We're looking at mechanism. Mechanism also represent process. There are two mechanisms that brings about variation. One is crossing over during Prophase I, and the second one is the random arrangement of chromosomes at the equator during metaphase 1 and metaphase 2. These events brings about variation. The event of bringing in variation starts with Prophase I during crossing over, and it continues with metaphase 1 and 2. All right. So let's proceed on and let us still explore some questions that involves logical reasoning. So here we have the following. Which phases, here it's not talking about mechanism or process, it's asking about the phases of meiosis, okay, that can bring about variation within a population. So which phases of meiosis brings about variation within a population? So the answer will be prophase 1 and metaphase 1 and 2. So you can't mention crossing over and random arrangement of chromosome because the question is not asking about mechanism or processes. It's asking about phases. So look at the word phase. Is there any word in under meiosis that is actually similar to the word phase? And here we are, we have prophase and then we have metaphase. You can actually find the word phase there. So writing crossing over will be incorrect because here we're not talking about process. Crossing over is a process, it's not a phase. It actually occurs in prophase. So the question here is talking about prophase, talking about phases. That's why you need to concentrate on these two. That's why the answers will be prophase 1 and my metaphase 1 and 2. Right, very good. So, as we draw the curtain, let us look at some important facts about meiosis. So, here, since meiosis 2 took place in two cells, there will be four daughter cells at the end. Now, these cells will be haploid automatically, and they are also automatically genetically different from each other. Now we need to know meiosis causes gametogenesis. That's why I was using it previously. Remember, this one is a revision exercise, and we have already done gametogenesis. And gametogenesis is the formation of gametes. So, in line with the question, why siblings are not alike, or why siblings don't look alike, and we traced it to meiosis, you can actually tell why meiosis causes siblings not to look alike is because meiosis introduces genetic variation during gametogenesis. So we have the role of meiosis, mitosis, and fertilization in human life cycle. We have the two parents, so they are both 2N, meaning they are diploid, and then they will undergo gametogenesis caused by meiosis. In the case of the male, gametogenesis is called spermatogenesis, which will lead to the formation of sperm cell. And then in the case of the female, gametogenesis by meiosis is called oogenesis, and it will lead to the formation of haploid gamete called ovum. The two gametes will fertilize, and then it will lead to the formation of diploid cell, something we call a zygote, and then the zygote will now undergo mitosis, which will lead to growth. Mitosis brings about growth, and then this will carry on throughout the life of the organism until they reach sexual maturity. Hence, we can say meiosis causes gametogenesis, and there are two types of gametogenesis. The one in males are called spermatogenesis, and the one in females is what we called oogenesis. It leads to the formation of an ovum. What is the importance of meiosis then? It leads to the production of haploid gametes. So the halving effect of meiosis, it prevents the doubling effect of fertilization. Hence, it ensures a constant chromosome number from one generation to the next. Secondly, It's a mechanism that introduces genetic variation, that is, genetic differences within a population of an organism, and it does so through crossing over, and it further does so through the random arrangement of chromosomes at the equator. Now, in every natural biological process, there will be some disorders. For example, humans are born with two legs, but then in a population of people, you find out that some people are born uh, without legs. No, everybody is born to hear sound with his or her ears, but then some people are born without the ability to hear sound. They are deaf. Same applies to blindness and uh, being disabled. Okay, so meiosis is also one of those many biological processes. Hence, there are sometimes some disorders that may occur. Under this curriculum, we're going to look at only one disorder. So here we have what we call abnormal meiosis. So abnormal meiosis, it is caused by non-disjunction. So non-disjunction of chromosomes at position 21 during anaphase 1 or 2, specifically in humans. So remember, as we started with the process of meiosis, we realized that you start with one cell, you end with four. You start with more chromosomes, you end with half the number. So here we started with two chromosomes, and each cell at the end will have one each. But how will you identify non-disjunction? So you can only know right from wrong, or sometimes you can know what is wrong by looking at what is right. So for us to know what is wrong, let us look at what is right. So the first diagram represents uh, normal meiosis. Abnormal means there's a disorder. So this one is in a healthy cell, and this one is actually a normal meiotic process. Here we begin with one cell, we end with four. We started here with two cells, and at the end of meiosis, we started with two chromosomes, sorry. So one cell containing two chromosomes. At the end of it, we're going to have four cells containing one chromosome. So the cell we started with is described as diploid, and you're going to end up with four haploid cells that are not similar. So that is how a normal meiosis should look like. But then compared to this particular diagram here, you will realize that there's something wrong. Over here, we started with one cell. Yes, we ended with four cells. But then here, each cell, the cell we started with it was diploid. We started with one diploid cell with only two chromosomes. We were expecting to to get one or to find one chromosome in each cell, but unfortunately, that's not what happened. It seems that instead of all the four cells receiving one chromosome, one cell received nothing. And then realize that the reason why it did not receive anything is because the chromosome it should have received is actually in the other cell. So instead of the third cell receiving one chromosome, it received two. So what happened is written there. Okay, that's the third cell received an extra copy of chromosome uh, inside it, and the cause can be traced to non-disjunction. So we are still looking at abnormal chromosome. An abnormal chromosome is caused by non-disjunction of chromosomes at position 21 during anaphase 1 or 2 in humans. So that is it. Now we need to look at what non-disjunction is. So let us ask the following question. What is non-disjunction? And the answer is as it appears on your screen. It is when homologous chromosomes or sister chromatids fails to separate during anaphase 1 or 2. That is all that you need to know about the meaning of the word non-disjunction as we prepare for the exams. Once more, it is when homologous chromosomes or sister chromatids fails to separate, or it is when they do not separate. It's when the homologous chromosomes or sister chromatids do not separate. The word fails here means do not separate during anaphase 1 or 2. So at the beginning, as we're doing our analogy, a replicated chromosome can change into an unreplicated chromosome, and that can only happen through anaphase 2. And we said during Anaphase II, spindle fiber contract and pulls chromatids, causing the centromere to split or break, and this causes sister chromatids to move to the opposite pole. However, during non-disjunction, that event fails to occur. Now, let us look at the effect, the result, or the consequences of non-disjunction. It results in the formation of abnormal gametes with an extra copy of chromosome 21. That is, there will be three chromosomes instead of two. And then a fusion or a fertilization between an abnormal gamete, that is a gamete with 24 chromosomes, and a normal gamete, that is a gamete with 23 chromosomes, may lead to a condition called Down syndrome. Down syndrome, also called trisomy 21. Why do we call it trisomy 21? Because on the human karyotype, if you go to position 21, instead of it being homologous, having two chromosomes, you'll find out that there are three chromosomes there, and the cause is non-disjunction. Non-disjunction caused it, and it will lead to a condition called trisomy 21. But according to the curriculum, it will lead to a condition called Down syndrome. So this is a female with Down syndrome, and as it should be. So how will you know what is abnormal in a person or in an organism? For you to know what is abnormal, you have to compare it to the normal. So we have on your screen the normal human karyotype. If you check from position 1 to 22, they are all in pair. We have one pair at each position. But then if you compare it to the person with Down syndrome at pair 21, you'll find out there are three chromosomes there, and it was caused by non-disjunction. The abnormal zygote occurred that way. The event happened that way due to, or the circumstances occurred that way due to non-disjunction. Hence, non-disjunction is the cause of abnormal meiosis. So if you compare the two, you can tell. Look at the normal one, position 21, we have two chromosomes. But then if you compare it to the one with Down syndrome, you discover that at position 21, there are three chromosomes. Hence, we call it trisomy 21. The word tri means three. That is three chromosomes at position 21. So that is a normal. This person, this zygote is normal, and then this zygote is abnormal. It's abnormal because at position 21, there are three chromosomes there. All right, so same applies here. If you actually look here, you are able to tell the gender of this person. That is a male, and this person is a female, and we know the reason why they are males and female. But if you check the male at position 21, you will discover that there are actually three chromosomes there. So it means this zygote will actually develop the condition of Down syndrome, the symptoms of Down syndrome. And then, but then if you check the female, it seems the female is normal, but the male is not normal. And the reason because there are three chromosomes at that position. So that male has Down syndrome or trisomy 21, three chromosomes at position 21. Okay, so this is an image showing the symptom of a person with Down syndrome. That is trisomy 21. All right, so to conclude on the presentation, to bring the whole presentation to an end, you need to know how to describe the following, which is very important under this topic, that is non-disjunction and how it leads to Down syndrome, and crossing over, the process of crossing over of homologous chromosomes. Not forgetting the process of interphase also, you need to know it. Then the following must also uh, be paid attention to. We need to also focus on the following: similarities between mitosis and meiosis, as well as the differences between mitosis and meiosis. And you can also top it up with the differences between meiosis 1 and meiosis 2. Well, this brings the whole presentation to an end. Thank you very much for joining me on this presentation.